what I'd like to do is talk about our most recent model. And we've been working on trying to reverse engineer the mouse embryo in particular. Basically, computational embryology is what we're focused on, and we're wrestling with the embryo as a complex multicellular system. So just a little background for those that might not be all that up on developmental biology, but in terms of looking at vertebrate embryogenesis, the basic vertebrate body plan is laid down very similarly across most, or essentially all vertebrate species. And back in the 1800s, a Darwinist, Ernst Haeckel, has this very famous portrayal where he was able to portray the natural selection of basic anatomical features in the embryo. And the cool thing about this is with that pattern, you can actually see things unfolding using very conserved logic, going from early development up through the different anatomical configurations you see across vertebrate species. That's actually come under attack by the folks interested in intelligent design, and it invoked cries of outrage from the creationists. And so topic was covered in Nick Hopwood's book, looking at the patterns of development and how it's come out of favor with certain communities. I thought intelligent design, though, is kind of interesting because that's ultimately what we're trying to do with the computer. And what we know about development is that these conserved pathways have a number of key principles of development. These key principles cover all sorts of organisms. Some of the toolbox, the molecular toolbox that's used, say, in mammalian development or vertebrate development, is using the same kinds of tools that a Drosophila uses, a fly uses, except the toolbox is put together in a different way and the tools are used in a different way. But the key principles of development are covered in most textbooks, such as the most common Scott Gilbert textbook in developmental biology that most undergraduates would use. And they talk quite a bit about the basic processes of development. What are the key processes and how are they shared by different diverse groups of organisms? So computer modeling can help reveal this complex spatial dynamics and the multi-scale complexity of the embryo as a self-regulating developmental system. And that's what I want to focus on today. So self-organization is an important principle. Just a little bit of history. I wanted to cycle back to 2009 while I was at the EPA, just recently joining the EPA, actually. One of the first things we did was to host what was called the first international workshop on virtual tissues. You can see James in there. There he is. There's me, a number of other people you might recognize. So these were folks that came across the globe. It was a joint workshop between the European Union and the United States, and basically brought people that are modeling different aspects of biology for different purposes together. And at the end of that, we wrote a sort of a position paper that summarized the workshop. And I actually was scribing the workshop, and I was sort of monitoring everybody's talk, and I wrote a poem at the end of that. It actually was included in this publication, but just describing sort of the events at that. But the cool thing about that workshop is, first of all, that's where I first met James, and we got to talk a little bit about the type of modeling that was important for virtual tissues research. And the other thing was there was this real neat presentation that struck me from a group at Endogenics, Tim Anderson, Richard Newman, and Tim Otter's work. And they were working with cell agent-based models. And in particular, what they talked about was anatomical homeostasis in a self-regulating virtual embryo. And in their model, each individual cell is shown. The cells are communicating through computational language. You'll see that shark bite takes a group out. And now the 23 cells, I think it was 23 cells, they start saying, okay, something's wrong, and they recompute. They're processing information on growth, adhesion, cell death. You'll see cells growing. And ultimately, what's going to happen is it's going to get back to the starting configuration. The group that was working on this was actually funded by the Virtual Soldier Project, and they were interested in the communication lines that were required between individual agents to maintain a reasonable size of a particular, say, brigade. And they wanted to take some of what they were doing and applying it to biology. And so this was a really cool presentation. It's something that I like to show pretty much in any presentation I give, because it shows the power of computational modeling. And in this case, this is an evolutionary model. So for this type of artificial life model, it's a phenotype-based protocol. And what they're basically doing is looking for the sets of rules that would be exchanged between these cells to self-regulate to the starting configuration. So that process is of self-organization. Another term for it is autopoiesis. And in development, there's an important concept known as canalization. So when you have a stress to a particular cell or group of cells, such as what you saw in this model, canalization is the ability to recover from a perturbation. And those two principles are very important when you're studying embryonic development, because embryonic stem cells do self-organize into different organs and tissue rudiments. And we know from our studies with chemical exposures that the system can recover from stresses at certain levels. Once you hit a stress point where recovery is no longer possible, and you get things like birth defects or other problems with development. But with these types of cell agent-based models, so again, the computational unit is the cell, and that's also the biological unit. So it makes a lot of sense to do cell agent-based modeling to try to recapitulate developmental processes. And over the years, we found that these models actually do have some internal intelligence. So they're smart models. And the reason they're smart is because we tell them what phenotype, in this case, what phenotype they were looking for. Now, there may have been 1,000 different combinations that never self-organized to the starting configuration of 21 cells. But in this particular case, the sets of rules allowed that recovery. And one of the things we've seen with our work on modeling through CompuCell 3D is that these models are actually, they are smart. They do things that are not coded into the model, and they produce effects that you can see evolve in the computer simulation and trace that back to known biology. So we've actually been really impressed with how smart some of these models can be if you give them the right amount of information. And with that said, you know, hypothesis generation is an important application of this work. So using these agent-based models to analyze these complex systems and begin to experiment with limitless what-if scenarios. So one could, say, start removing specific cells or specific groups of cells or changing patterns of interactions between these cells and generate phenotype-based predictions. Now, we call these types of phenotypes that are generated in silico, we call those cybermorphs. So group's been working with CompuCell 3D over the week. I don't need to give much background, but again, CompuCell 3D, in our case, in our handling, has been treating individual cells as autonomous agents. Of course, for development, we have to define the different cell behaviors, regulatory signals, physical properties that go with the system, and allow it to calculate the evolution of the system in a multicellular way and in a particular spatial setting that we have to sort of design to fit the purpose of interest. The system aggregates molecular control logic and produces emergent phenotypes, again, cybermorphs. And the question then, of course, is validating that your model is producing the phenotype that can be experimentally validated, and that's a real challenge here. But they provide fully computable code-to-dynamic simulations for prediction, and the practical applications that we've been using over the years has been in trying to decode the toxicological blueprint of developmental defects. So development, as you know, is generally described clinically in 3 trimesters. That first trimester has the period of development that the embryo is most susceptible to changes, either genetic changes or environmental changes or combinations of the two. It's the period that the embryo is most sensitive to structural birth defects. And there are well-devised testing guidelines from the Organization of Economic Cooperation and Development. Test Guideline 414 tests that embryo period in rats, pregnant rats and pregnant rabbits for different chemical effects. And I just threw a couple of percentages from the CDC in terms of the rates of different developmental defects that could be affected during this period. These adverse birth outcomes include preterm birth rate, that's 10% of births in this country, low birth weight babies, 11%, malformed babies, 3 to 4% of the live births, mortality, almost up to 1% of newborns, and functional deficits, which include things like learning deficits and things. Actually, it's almost at an epidemic level in this country. One in 6 or 7 children, by the time they're screened for school age, have some type of a functional deficit. It could be associated with a very minor malformation, or it could be something that you don't even notice. So we have a lot of reason to try to understand during this period of time how the embryo develops, how it's affected by chemical exposures, and there's just some of the primitive cell behaviors that are important in organizing the embryo. So cell growth and programmed cell death, differentiation, and functions such as extracellular matrix production and remodeling, cell motility, cell adhesion, molecular clocks, pattern organizing systems, different genetic signals and their responses, and of course, extracellular matrix biology. So we've been working with a lot of folks, some from James Lab, some from other groups over the past 10 or 11 years. And this is just a sample of some of the models that have been developed with CompuCell 3D. Now, the somite model was directly out of Jim Glazer's group. Our first model was looking at vascular development. We've got models for the limb bud, for palate fusion and cleft palate, for genital tubercle formation and sex diversification. Some of the newer things we've been working on, the Epiblast model, neural tube closure, blood-brain barrier morphogenesis, yolk sac morphogenesis. Jim Glazer and his team has been working on corneal modeling. So we include eye development as well. And the things like heart development, liver development, kidney development, and testicular development are things that haven't yet been approached, but should be things that are important because these are systems that are commonly affected by drug or chemical exposures during pregnancy. So there's just a short review that we wrote on what we call the virtual embryo. And this is sort of a way of integrating all different systems. Now, we're not trying to build a computer model that's going to spit up on you or soil a diaper or cry or throw something at you. But what we do is we focus on different systems that have critical requirements biologically to occur at a certain time in development. And we model those as modular systems. And then eventually the whole thing will come together as an ensemble. What I'm going to talk about for the rest of today is our most recent model, which was just published a couple of weeks back, on focusing on the Epiblast and basically engineering a computable Epiblast in CompuCell 3D for in silico modeling of potential effects of chemicals and drugs on early development. So why is the Epiblast important? Well, first of all, if you're familiar with what's going on in the world of stem cell biology, embryonic stem cells in culture most closely resemble the pluripotent differential potential of the epiblast as the embryo undergoes a process known as gastrulation. And you could actually put stem cells in culture and they will aggregate into these so-called embryoids. So these are arrangements of cells that can differentiate spontaneously into things like heart, blood cells, a number of different differentiated systems. They don't necessarily form normal-looking embryos, but they have the potential to give rise to all the cell types that essentially will form an embryo. And the embryo is a lot smarter than the stem cells because it understands how to put everything together and give you limb buds and branchial arches and a heart and a developing brain, the anterior to posterior axis. That axis is actually established in the epiblast. And this is what a human epiblast looks like. This would be about the third week after fertilization, usually about 3 weeks before a woman might be diagnosed as clinically pregnant. And the hallmark feature of the epiblast is primitive streak formation. So this is a feature of all vertebrate species, or most vertebrate species. The formation of the primitive streak is something that's been very heavily studied by developmental biologists for reasons I'll mention. But in particular, what it does is it positions that anterior-posterior axis. So at this point, the tissue is committed that this is going to be the head region, this is going to be the tail region, and all of those structures will come together and eventually look like this a few days later. And the other thing that is important about the primitive streak is that the mesoderm, which is the tissue that forms most organ systems in combination with different epithelial cells, but that is actually formed at this time in development. And so you have two things going on that's important. Number one is the establishment of the anterior-posterior body axis, and secondly is the formation of the mesodermal population. So the model that we built, this was a 3D model in CompuCell 3D. We've done lots of 2D models, which are a lot easier to work with than the 3D model. But I'm going to show you what it looks like and then sort of go through some of the basics and what you do with it. But here's the formation of the primitive streak. And what's happening is the cells are migrating through the primitive streak, and in between, at this point, it's a two-layered system, an outer and an inner layer, and the mesoderm forms in between. And I'll run that again. Richard Spencer developed this really cool transparent rendering of the system where you can see through the epiblast cells as they're migrating through the primitive streak into mesenchyme and migrating out towards the periphery. And there's a very famous quote from Lewis Wolpert. And he says, "It's not birth, marriage, or death, but gastrulation, which is truly the most important time in your life." So without gastrulating, you wouldn't be here, and you wouldn't be able to get born, married, or die. And so, you know, it is an important time. It's a period of time that the stem cells in the epiblast are differentiating and receiving information on what they should be doing in the next stages of development. And that's what we're trying to model here. Okay, so here's what's important about the simulation. So in the epiblast itself, so there's a primitive streak. Again, that'll be the anterior side. This is made up of pluripotent epiblast stem cells. Again, these are the cells that, if you culture stem cells, they tend to reflect mostly the molecular character of the epiblast at this stage. What ends up happening here is that these cells are migrating by epithelial, it's a developmental process. So they're moving towards the primitive streak where they undergo epithelial mesenchymal transition and then migrate inside between the outer layer and the inner layer of the structure. So you go from a 2-layer pancake to a 3-layer pancake called a bilaminar embryo to the trilaminar embryo. And that is the result of gastrulation. And you can imagine that if you're a cell further away from the primitive streak, it's going to take you a longer period of time to get there. And you're going to enter it depending on where you're coming from. So if you're coming from this side of the epiblast, you're going to tend to form, you're going to go through the primitive streak where it's going to be anterior. If you're on this side, it's going to be going through the primitive streak where it's going to be more posterior. So that's an important concept. And this is one thing that really is an appeal for the agent-based modeling approach, because the position of a cell, positional information is very important. And you can track all of that individually, cell by cell, step by step in CompuCell 3D. And as I note here, it's a key determinant of mesodermal fate. Okay? So where and when the cell passes through the primitive streak essentially determines what that whole lineage of cells is going to contribute to. Now, you can mess this up. You can kill certain cells. You can delay certain cells. You can have different effects on it, and that's going to have a significant effect on the next stage of development. So this is what the CompuCell model looks like if you don't use the transparent rendering that I just showed you. This light blue layer is actually extra embryonic ectoderm, which sends the signals that start everything. But as the model starts, now this is a one-shot model, so the one epiblast layer, it's not regenerating itself. It's giving rise to mesoderm that's migrating into the primitive streak and then out to the periphery. And what we're doing in the CompuCell model is specifying different regional fates of the mesoderm. So these green cells are going to be cells that form the notochord. The red cells are going to be forming what's called paraxial mesoderm. The purple cells are forming different regions of what's called lateral plate mesoderm. And so as you see the one-shot model, there's the primitive streak here. The epiblast is consumed. Now in real life, it would be regenerating itself. When we do that, you'll notice that the boundaries are not growing. So if we have the epiblast continuing, the mesoderm they just grow up, they just pile up on the periphery. So that's one limitation with this model. It's not, the model is not growing as an embryo would be growing. It's just showing us the patterning that's happening in a one-shot consumption of population of epiblast cells. There's just another view there. Just looking at the mesoderm, not looking at the epiblast, so that's made transparent. The yellow layer is endodermal cells, and these mesoderm cells, again, they're migrating in between the outer blue epiblast and the inner yellow, what's called hypoblast. So we've worked with the code for a couple of years to try to get this to represent what's normally happening. Now, even though we're modeling what looks like a human embryo, we're getting our cues from the mouse. And there was a nice study published by McDowell et al. In Cell in 2018, where they used light sheet microscopy to look at the mouse embryo and map out the different fates of mesoderm. And so here are the populations that are in their model. So the green there is the green cortomesoderm, the red is the paraxial mesoderm, the purple, you don't see it on both sides, there's the lateral plate mesoderm. And what we actually are very interested in is this posterior population of the lateral plate, which we color orange, because that's the population of cells that's going to give rise to caudal structures in the embryo, but more importantly, most of the blood cells that are going to form for that individual. And we've been interested in modeling blood development. So again, the one-shot model, which we call Epiblast stem cell agent-based model, or ESABM, that's been tuned specifically to recapitulate the pattern of mesodermal cell specification in the mouse embryo. And it does a pretty good job. So our paper was reviewed by at least one very well-known developmental biologist. I know pretty much guess who it is. It's pretty much the guy that has characterized the primitive streak. And we followed lots of instructions from our reviewers in terms of what was right and what was wrong with the model. And by the time we got to this point, and I think our other reviewer was actually Katie McDowell, they really liked the model. So we got lots of accolades from our reviewers, and that's now published in Reproductive Toxicology. Okay, so to get to this point, as you probably are aware, it's a very complex workflow. So it starts out with problem formulation. We were interested in modeling gastrulation because that's the most important time of your life. We know that defects in gastrulation are very hazardous to subsequent development. So Nancy Baker and our group developed some really neat and important literature mining tools that basically allow you to compile relevant information, extracted information for the questions you're interested in, build ontology maps of how the system is controlled. Then we go to our CompuCell 3D framework. We start exercising the model with looking at different cell types, such as the mesodermal cell types, the signaling gradients that are responsible for formation of those different cells. And I'll talk about that in a moment. I showed you the rendering of our synthetic Epiblast prototype, which has been through several renderings. And we can now execute these simulations, analyze output parameters such as how many cells are forming over time, how many cells of different mesodermal fates are being produced under different conditions, and start doing a sensitivity analysis by different genetic perturbations. So we've got lots of signals that control the model that are genetic in origin, and we can start perturbing these signals. Now, in the real world, people will knock out a particular gene in a mouse and look at complete knockouts or heterozygous knockouts. In a computer model, you can do this electronically, and you can actually run titrations to find out what that critical perturbation is going to be before you start seeing a defect. The validation of the model outputs is important. Again, we talk about cybermorphs as in silico phenotypes that we can compare to different literature studies where they've knocked out the different genes, say in a mouse, and have described potential phenotypes. So we can compare the results from our in silico cybermorphs to the real world. And then as a practical application, one way of using these models is to operationalize the system to look, say, at chemical stressors. So we've got databases, the ToxCast database, for example, that has millions of dose response curves for thousands of chemicals on thousands of potential molecular targets. And we can start incorporating some of that database information into these models and make predictions quantitatively of what might happen. So are you going to get, say, 1 out of 1,000 disrupted simulations, or you're going to get 10 out of 10 disrupted simulations? We've done that type of thing over the years with some of our other models, and actually the predictive power of a CompuCell 3D model has been pretty good comparing to the real world. Not perfect, but pretty good. All right, so what's controlling the network? It's actually a little bit more complex than this, but this is a simplified version of the network. So if anybody is interested in developmental biology, you'll be familiar with these signals. These signals also show up again for a lot of disease processes such as cancer. But we're looking at the interactions that are described for this time in development for bone morphogenetic protein 4, nodal, Wnt, lefty, fibroblast growth factors 4 and 8. There's some interesting sociology that went into this, because when we first submitted our model, we didn't include nodal. It was mostly driven by these other factors. And one reviewer, which again, I think is the sort of inventor of nodal, said it would be unconscionable to model this stage of development without nodal signaling. So we spent several months incorporating that into the model. And it was really very well worth doing. So, nodal signals, it signals on its own. It induces the expression of lefty, which actually is a direct and an indirect feedback inhibitor of nodal as well as BMP. Wnt3 is the classic biomarker for primitive streak formation. And that's actually regulated by fibroblast growth factors. And I'll talk a little bit about CDX genes, because this is an important aspect. So fibroblast growth factor 8 controls cell migration, but fibroblast growth factor 4 as well as 8 controls what's known as the homeobox clock. And it's the homeobox genes that specify mesodermal patterning. So CDX is kind of the master gene of all of that, and it's regulated by fibroblast growth factors as well as things like retinoic acid and Wnt itself. So that's an important part of this. The signals themselves are shown here. This is what they look like from the top view as they're evolving. There's a primitive streak there. There's lefty. Lefty is expressed in what's known as the anterior visceral endoderm, AVE, and nodal is very important for patterning the whole system. And this was our sort of ontology table that describes the signals, what their targets and responses were, and the consequences of those actions and references that support that. Okay, so now just looking down again at the top of our transparency model here, there's a primitive streak, anterior left, posterior is right. There's the cell field from the simulations I showed. So we can map out all these different signals, but start doing electronic knockouts. So turning off certain signals and looking for effects. And what this is showing is simply that there is crosstalk that's built into the model that's derived from, the model self-organizes, but we derive that information from the literature. So for example, we know that, as I said, lefty inhibits, it's an antagonist of BMP. And so when you take lefty out of the system, here's the ring of BMP4 expression in the extraembryonic region, and it's pretty uniform throughout without lefty. Lefty is expressed here. Where is it? Well, in the AVE, there's lefty. And you see that in the model, it's actually suppressing BMP4. So, we can do that kind of gymnastics and play around with the system and look in an array of models. So each of these models is basically one simulation. And for BMP4, this ranges in the model from complete loss of BMP4 in the model to reductions of 75% and 50%. The isomorph is the baseline, the optimized model that gives rise to the McDowell pattern of mesoderm. And we also looked at hypermorphs. So what happens if you have too much signaling? And you could see, just focusing on this posterior mesoderm, you could see that it was clearly dependent on the amount of BMP that's present. And that's known from the biological principle that the BMP is having an important effect on patterning the posterior mesoderm. We could do the same thing for nodal. And you'll see basically a similar type of an effect because BMP and nodal are working together. So going from no nodal to ludicrous amounts, not as strong of an effect as the BMP, but you can see that it does have some effect on, in this case, at the highest levels, we're seeing less posterior mesoderm. So what we call anteriorization of that embryo versus posteriorization. lefty again, and FGF8. So FGF8 is cool. FGF8 is required for cells to migrate away from the primitive streak. In the real world, if you knock out FGF8 in a mouse, the cells pile up around the primitive streak. And you could see that that's happening here as well. Over that period of simulation, not all of the one-shot epiblast even goes through the primitive streak, and you're not seeing cells migrating out to the periphery. And then the question can be asked, how much FDF does the system need to look normal? I don't know why it's jumping. But you can find out sort of what that critical level is, where you start seeing disruption of populations of cells that you care about, such as the posterior mesoderm. And you can map that out. I'm not going to go through this, but this is just looking at the quantal distribution of these different cell types for the mesoderm as a function of how much signal there is from a complete knockout to a ludicrous amount of signal, where one is the control. And you can see that you can start making statistical arguments about how much in the model is critical for normal development. Okay, so I talked about the homeobox clock and specification. And you've probably heard the term homeobox genes. Some of you probably know a lot more about it than me, but the homeobox genes are very important for setting up the pattern of development. Mammalian species have 4 clusters of homeobox genes, HOX A, B, C, and D. Each cluster has paralogs of up to 13 different homeobox genes from 1 to 13. This was first discovered in Drosophila. And what was really striking about these homeobox genes is that the order of these genes on the chromosome from 3' to 5', and that's how they're numbered. That's the order that they're expressed developmentally and anatomically in terms of the anterior-posterior axis. So the different homeobox genes are nested based on combinations of genes. So you may have several homeobox genes expressed in a particular cell type, ranging from just maybe HOX A1 up through combinations of HOX A1 through A9, for example. And I mentioned earlier that in our gastrulation model, cell position is important. And this is where we can really make some headway in using these models. So you think of it as a clock. So here's 4 homeobox genes, HOX D4, D9, D11, D13. So 4, let's look at 4, 9, 11, and 13. And you think of it as a clock. So again, the order is a developmental time measure. So if you look clockwise, that HOX D4 will be first expressed out of those 4, and it will generally stay expressed throughout because one homeobox gene will have an influence on the next. As HOX9 is activated, maybe at 3 o'clock, that'll continue, and so forth. HOX D11 at 6 o'clock, HOX D13 at 9 o'clock. And so if cells are coming through the primitive streak early, they're going to be primarily expressing these more anterior homeobox genes, and that's going to specify their positioning across the embryo. And as you progress, you get these nested patterns and more complexity in the homeobox patterning as the clock continues. Now, the cool thing about it is that the cells are measuring time by this progressive activation. But this sort of colon area, as it's called, is set. So the pattern of homeobox clustering, or sorry, homeobox expression, or what's known as the homeobox code or the HOX code, that's set as the cells pass through the primitive streak. So the clock is winding down through the epiblast, and as the cells migrate through the streak, they inherit whatever combination of box genes happen to be active at that time in development. And then they carry that forward in their development and in their lineages. So as they give rise to different cell types, as the embryo develops, they tend to obey that regional specification. Okay, so that's complex biology. It's more complex than I'm describing, but you can model that. And that's shown here. So we can show that stereotypic activation of homeobox genes in time and space. So there's our isomorphs. And in this case, there's the 4 homeobox genes that we were modeling. So the D4, D6, D11, and D13 in different colors. And I'll go back to the schematic. Okay, so again, 4, 9, 11, and 13. And you're going to see that the 4, so we set the autonomous clock in the code. And as the cells migrate through the primitive streak, they're colored by whatever the latest homeobox gene would be. So they'll start out in that blue color, then a green color, then a yellow color, and then a red color. So we'll go back to the plots, and here you'll see there's blue, then the clock switches to yellow, and then finally red. Now, I had mentioned that CDX is what's controlling the progression of the Hox clock. So fibroblast growth factors will speed up the Hox clock, and say retinoic acid signaling will slow it down. But they work through CDX. And when you knock CDX out in the literature, you got big problems. And here you see that the clock is not progressing because it's not interpreting the signals that the cells are receiving. So again, with this type of thing, we can quantitate the numbers of cells in each class of homeobox gene over time and what the result of perturbations of, say, fibroblast growth signaling might be on the system. And here is that same type of a plot showing the effect of fibroblast growth factor disruption, going from, this is FGF4, this is FGF8, but you can see that as you increase the amount of fibroblast growth factor, you progress through those 4 homeobox genes in collinearity. And that would be the normal, and this would be predicted by if you have too much FGF signaling, or if you have not enough FGF signaling, you'll simply just sort of stall. So we can incorporate into our network that type of interaction. I didn't talk about retinoic acid due to time, but they work together as a mutually inhibitory system and have effects on CDX. And there are chemicals and drugs that can have effects on either of those and potentially affect morphogenesis at this stage. So as we start thinking about how we're building ensemble models towards a more fully computable system, you can use CompuCell 3D not only in development, but in any system in 2 different ways. So let's forward engineer. So suppose you know what a molecular mechanism is. So a particular perturbation, whether it's a genetic defect or say an enzyme that's been inhibited. The question, of course, then is how far can these types of agent-based models take us to hypothesizing what a phenotype, where your induction is introduced into the model and the elicitation is the free parameter that you're looking at for phenotype. Or you can reverse engineer the system. So suppose you know a particular phenotype, such as say a birth defect. How far can the agent-based model take us to inferring a mechanism? And actually, I think probably the truth is somewhere in between. It's got applications both ways, and the success of these models is really dependent on the amount of biology that we put in. So just 2 summary slides, one on lessons learned and one on lessons unlearned. So computer modeling of key events in any system, but in particular the embryo, is a crucial and challenging effort. Although I think we've learned over the years that it's feasible in the CC3D modeling environment. And James is always very good about saying CC3D is not the only type of modeling system, that modeling platform available for looking at agent-based modeling. But in my mind, in terms of the flexibility of the model and the ability to really spatially constrain your system to phenotype-based protocols, I think it's a very useful application. It's got some limitations, as we all know. It does some things better than others, but in terms of modeling the system, you can model systems with as many as 13 or 14 different genetic signals interacting and begin to make predictions that you couldn't do in the real world. So that's what the actual practice is using this. And extraordinary efforts and amounts of time are going to be required to build these types of effective models that can be used in practice, either for mechanistic simulation in a multi-scale system or for hypothesis generation. Small steps forward are useful. What we really want to try to do is stimulate interest in these types of virtual tissue models and their practical application for multi-scale modeling and simulation. And so that's why I was very happy when Hayden invited me to give a talk to the group. I think this is certainly a population of individuals that can really take this type of modeling to the next level. As I mentioned earlier, we're not trying to build something that's going to cry or spit up on you or soil a diaper. And just to get to a fully computable ensemble model of the embryo, it is a blue sky vision. Can start really being used to test real world data that you can build into these artificial life streams experimentally, just because of the sheer complexity and how you can manipulate these models. And you're not knocking out a pathway or a gene. You can say what amount of that signal is required for normal development before you start having problems. And so that really is a way of allowing us to begin to bring some attention to the value of these models in setting what are critical phenomena in terms of, say, a birth defect. And of course, these types I mentioned, they allow you to simulate scenarios. You can begin to localize these critical phenomena in complex self-organizing ways. We can look at combinations of genetic perturbations, so not just one gene knocked down, but combinations of genetic effects. And we know even from, say, the homeobox literature that you have to knock out more than one paralog in a cluster. So the combination effects are really important in terms of what these models offer. The question is convincing users and stakeholders that they're reliable. But there's some lessons unlearned. This is a pretty cool example of intelligent design of what's called xenobots that are formulated by artificial intelligence and then handcrafted from biological cells, such as frog stem cells, embryonic stem cells. There was a spotlight of Mike Levin's work in this called Code to Creature. What they're doing is they're modeling or designing life, artificial life in the computer, and then they're building it, in this case, using frog stem cells, the brain. I'm not sure what the green cell type was, but they make predictions in silico, just like engineers working on the 747 built the 747 in the computer before they ever built the plane and tested it for all sorts of parameters. And so, this is sort of that code to creature view of design it in silico, build it in vitro, and apply it in vivo. And so the link to that paper is here. And then finally, just for the paper that I talked about, so Caitlin Barham, Richard Spencer, and Nancy Baker were the team that worked on this particular model. Caitlin had another commitment at this time, otherwise she would have given this talk. And she invited everybody that might be interested in learning more to certainly write to her. So we have their email addresses here. Richard is really a stellar collaborator. He's worked very closely with Mashek, Swat, and others at TJ Cito and Jim's group on various problems that have come up along the way over the years in terms of parallelizing the system. He's developed a virtual tissue laboratory system, which is a computational infrastructure where he runs these models, such as the transparent model that is shown in the upper left. And he can run them on the supercomputer at the EPA, and usually have to do it at night on weekends. Otherwise, people get upset at how much time it takes away from everybody else. And then Nancy has been our expert informatician on, she's worked on most of these models for the virtual embryo. So feel free to contact me if anybody wants to learn more. As James mentioned, I'm retired from the EPA, so technically I can't speak for the EPA, but still happy to collaborate with many folks that we've worked with over the past few years and are continuing to work with.
Yeah. Hi, Dr. Thomas. It was really great to attend this talk. I have more of a technical question. The model looks very computationally very complicated. There are multiple fields and thousands of cells, and there is all the model is also in 3D. So I wanted to know, like, what computer systems did you use for simulating this? Since I'm trying to build a model which has like 2 to 3 fields, but it's very like the runtime is very slow for my model. So I just wanted to know what systems did you use?
Gopal, I think a high-end gaming computer is probably the smart way to go. We've had people that have rotated through on different projects using very high-end desktops, high-end towers. Some have used Mac, some have used Windows, some have used Linux. Richard Spencer does our supercomputing work, and the throughput is pretty good on that. The one thing that we've learned is that the administrative aspects of running something on at least our EPA supercomputers are very different than what you'd run on your own laptop or a virtual laptop. And so the code that we use for the virtual models on the supercomputer is slightly different from standalone computers. And it's mostly in the rendering aspects. So that's the challenge there. I'm not the one that does the coding. I just, as everybody does the hard work, but you know, I'm certainly aware of when folks have had issues with things running slow. Our limbud model, we can run our limbud model to simulate 3 days of mouse limb development. But it takes more than 3 days to run the model. So it's running slower than biology. But I think, you know, about moving into quantum computing and, you know, really to thousands of times faster, it's going to be less of a limitation down the road.
Are you aware that some of the chemicals are triggered by mechanical forces on cells? That the mechanical forces go down into the nucleus and kind of rearrange it.
You know, that's an important question, because developmental biologists overall are genome centric. So genes control everything. And it's very clear from Mike Levin's work and other types of work, just exactly what you're saying, that electrical forces, mechanical forces actually can cue the nucleus and really be important. In Mike Levin's work on code to creature, you know, they are doing exactly what you say. They start from the physical environment and then manipulate the genetic environment that way.
Okay, I've got my name on a paper. They put it there. I don't know. I feel like it's an accident, but I don't know if you've heard of Bradley Alicia.
No, I don't.
Richard Gordon. Anyway, they've done some work on looking at the mechanics of cells and that sort of perturbation, and then what chemicals it produces, and they've got it mapped out. So that's in this most recent paper.
Mhm.
They've mapped out some of the forces to genetics in this most recent paper. Just as an example, limbs aren't created unless there's lungs. And lungs aren't created unless there's kidneys. And kidneys create amniotic fluid and the lungs breathe them in.
Mhm.
And that's sort of how all that starts physically. So that's an example of sort of multi-system forces. Is interesting.
Yeah, which we refer to as a physiome. So you have a genome and you have a physiome.
Oh, physiome. Okay.
At the EPA, it's right now one of the big issues is thyroid function during development. So the thyroid connects to the liver because the liver metabolizes thyroid hormones, and the thyroid hormones target the brain. So you've got really the need for a multi-organ system of the liver, brain, and a thyroid, which I used to call the BLT model, brain, liver, thyroid. The placenta also needs to be in there too, because maternal thyroid hormones get across the placenta.
Oh.
But yeah, I think, certainly mathematical models and systems models are useful for that type of thing. And maybe CompuCell models can model the individual responses at the microtissue level.
It's going to take more than one model and one
Yep.
system to figure this out. That's for sure.
Yeah, and the ensemble modeling approach, I guess, is what we want to push.
Well, it starts off by cells being stretched, and then they respond by cramping. And if they cramp for too long, that gets the physical signal into the nucleus and squashes the nucleus for long enough that it rearranges some of the cytoskeleton. That's the theory.
Well, the other thing is the work on the virtual lung that was published in Science 10 years ago from the Wyss Institute. They showed that if you have an in vitro model of the lung, of the alveolar space, and it's just a static model, you miss a lot of the physiology. So when they start artificially stretching and contracting the alveolar space, just like what would happen in vivo, and have a circulating system associated with it, that macrophages actually will cross the alveolar space if there's the breathing mechanism, but they don't cross if the breathing mechanism is absent. And similarly, even in the intestinal models in vitro, peristaltic movements seem to have an important effect on the physiology of transport, of chemical transport across the intestine. So, I think your point's well taken. Unfortunately, at EPA, they're so genome-centric that sometimes that argument doesn't carry through.
Oh, well, it's just kind of obvious. If you squash a nucleus, it's going to rearrange its genetic material.
Mm-hmm.
I mean, there's a whole pile of literature on that out.
So is somebody modeling that?
Maybe that's what they need to do.
Well, James, folks can write to us if they have things that come up.
Okay. Well, thank you very much, Tom, for the great talk.